A diatom is a unicellular eukaryotic organism that basically uses sunlight to do photosynthesis, much like plants do. Major difference here is diatoms are single cells, plants are multicellular. In terms of their importance, they're quite important in the oceans and in aquatic environments in general. So they basically exist in almost any body of water that you can look at. They make up about 20% of what we call primary productivity in the ocean, basically the energy that's being produced in the ocean. And so you can almost say about 20% of the oxygen on Earth, come from these diatoms.
Anything that's less than 100 microns is going to be washed right through the mesh. But within the mesh, we'll capture anything greater than 100 microns. That could be large phytoplankton, chains of large phytoplankton, large diatoms. Diatoms range in size from 5 to hundreds of microns, so we'll definitely have a lot of diatoms. You can find Cattosaurus, you can find Pseudonychia. And these are ones that you can find in coastal regions and open ocean regions all over. I would say that they're more resilient in the summer to salinity, really high temperatures. So already we probably have a couple hundred liters. So I'll pull this in.
So diatoms are a really good kind of way of, if you will, to indicate whether a body of water is healthy or not, because technically if you have a region where you have enough nutrients, you're going to have enough diatoms growing, that means the whole food web is basically being fueled, because there's going to be zooplankton and copepods that are going to feed on these diatoms. There's going to be small fish that will feed on these, and krill that will feed on these copepods and zooplankton. You're going to have larger fish and so forth all the way until we have pretty much whales. And so having diatoms there kind of supports this marine food web. If you don't have diatoms and other phytoplankton groups, then you don't have a very productive ecosystem, which means you're not going to have, typically large fish and the things that we'd like to see.
When I work with diatoms, I mainly look at their morphology and their attachment, free living of bacteria with diatoms, and also observe all the morphology changes of diatoms. This is axonic, and this one we add bacteria, and this is the one mixed bacteria. The diatom need bacteria to survive longer. So here, this is a cyanobacter, and apparently it looks like a net around this diatom and trap them inside. And one time I just saw this cyanobacter squeeze it, smother the diatom, and the diatom actually break and release their inside stuff outside. Yeah.
But one of the emerging ideas that myself, as well as many people in the field, have been looking into is, for a lack of a better term, kind of parallel control. That is, interactions between diatoms and other organisms that are, not grazers and not just chemicals. For example, bacteria, viruses, other phytoplankton. So all of these interactions seem to be quite abundant, quite diverse. They probably happen at all times. As we speak right now, there's trillions of diatoms interacting with, gazillions of bacteria and other phytoplankton species. And they're exchanging stuff, they're fighting against each other, and a lot of this is really fascinating. One of the things that I've discovered early on in my career and we still work on that in my lab, is that a lot of the currencies or the chemicals that are exchanged between diatoms and some of their bacterial partners happen to be molecules that are identical to the molecules that plants on land use to interact with their bacterial symbionts. Yet, even though plants and diatoms are both photosynthetic, they are not related. They're completely on different parts of the tree of life. Diatoms are related to brown algae. Plants are related to green algae. And so very, very, very different trajectories, yet you see these very similar, elements between those two completely different ecosystems.
I'm working on the classifier part. So we have the condenser here. The sample will go through the sample imports here through these ports, and it will reach the flow cell, which is located right behind. And there is a laser inside. And once the particles, the plankton cells, pass through the laser, the scattering and fluorescence will be recorded by the detectors. This here, and the camera, when it triggers, it takes the high resolution images of every individual particles. Yeah. Oh, this sample was really amazing. A lot of stuff. This is a crude sample from the field. Yeah. So this detect a particle based on the gradient difference with the background, and once it detects it, it's basically draw a square that will be imaged. Yeah. Now it's capturing the images. But with the volume we sample, we can analyze, we can get their counts with machine learning expert. And basically we will say to the system, okay, this one belongs to this category. And we train the model. If you train the model with maximum number of images, the model will be doing excellent. Yeah, so we will be putting this information to a convolutional neural network where the images will be transformed to blobs, and the features will be extracted, like the shape, the volume. And based on those information, the classifier works. Oh, this sample is really cool. We have a Chitosaurus, some centric diatoms, a lot of them, and yeah, it's a diverse community. It's black and white. Yeah.
They're one of the few organisms on Earth that have evolved to have a cell wall that is encased in this silica shell. We call it silica frustule. And that silica is basically the same chemical structure as glass. And so actually when you look under the microscope at diatoms, they look like tiny pieces of glass that have a little bit of color, which comes from the chlorophyll that they use to absorb sunlight. And so they come in all of these various shapes and geometric, patterns that are very intricate, and they're very accurate to the nanometer scale, which is extremely small. I mean, the width of the human hair is much bigger than that.
So now I'm up moving through, and it's moving really quickly. So, oh, look at that one. That one's probably a Chitosaurus or a Skeletonema. If you just apply a little bit of, suction to that diatom, you can capture it in the micropipette tip, and it's like a very small volume of water. By getting one diatom, by isolating one diatom and then isolating all of the bacteria within its phycosphere, I'm getting the identity of the diatom and also the identity of the bacteria. With genomics, I can also get their genomic potential, so their capability and certain types of metabolism, and especially if you see complementing pathways between bacteria and diatoms, then you can assume that yes, they're like symbiotic or they're working together.
So the diatom has more than 20,000 genes. So in one experiment, you are testing actually 20,000 genes at once. And also in the bacteria as well. Of course, the bacterial genome is much smaller. So you testing like 5,000 genes or 4,000 genes. So this is really comprehensive view of the whole genome gene expression to just one treatment, be it a bacterium or a bacterial product. For example, vitamin B6 derivatives that we are discovered that are mediating signaling between the diatom and bacteria, and then you see how the diatom is responding to this.
So over here we have our system for programming this nutrient analyzer over here. And basically this is where I would set up all of my reagents, my standards. I would put in the concentrations, and then I would program in all of my samples so that it knows kind of what it's analyzing. All of this works to kind of determine, whether there's eutrophication, whether there's human pollution, any input of nutrients into the water column, but also just looking at basically what's in the water column and can it support life, basically. Diatoms generally are the dominant species whenever you have silicate in a system. However, other species of phytoplankton, a lot of dinoflagellates can actually produce toxins which can harm all marine life, and they tend to reproduce quite quickly and take over a whole ecosystem and disrupt this symbiosis and this natural sequence of events.
So diatoms are, obviously are everywhere. The world is changing. We have climate change. We've got ocean acidification. It's not very clear how these are going to affect diatoms. We know some things are going to definitely affect them. So for example, ocean acidification has been shown to limit the availability of some of the things that are essential for diatoms, like iron, for example. And that may impact their ability to grow in some places. My suspicion is what's going to happen is diatoms are not going to be wiped out by any means because of climate change. I suspect they will still thrive, but I think there will be changes in the diversity. So some species are probably going to lose, they're not going to do very well, and other species are probably going to thrive under these new conditions, and then they'll become more abundant. But a lot of these things are, things that we haven't really examined in enough detail to really know the answer to. But that's the general thinking in the field right now.